So our first speaker is Tierney Bougie, and she's going to talk about the dynamics of hybridization in hybrid zone between members of the Habronattus americanus subgroup. So,
all right, so thanks for being here. I'll be talking about hybridization dynamics in a hybrid zone involving Habronattus jumping spiders. Oops. Okay, so naturally occurring hybrid zones can be used for answering several evolutionary related questions. Commonly, we can use them to identify genotype-phenotype associations. So like here are a lot of bird studies actually. They're identifying the loci that correspond to certain phenotypic traits of these male warblers. But we can also identify new species through contemporary hybrid zones. So a common example of that is sunflowers and the homoploid hybrid speciation. But there are other mechanisms like hybrid trait speciation in heliconius butterflies. We can also use hybrid zones as a means for identifying drivers of reproduction, or reproductive isolation, whether that's physical traits or otherwise. As far as phenotypic distribution throughout a hybrid zone goes, we can describe them in a few different ways. So bimodal hybrid zones occur when hybrids are very rare, so you don't see them really at all, and they're heavily selected against. And then you have your two parent types at really high density and equally density too. Trimodal, where you have both parents at a higher density than your hybrids, but your hybrid is still noticeable in the hybrid zone. And then unimodal, where you have hybrids and both parental types that are present at equal frequencies across the zone. And this happens when you have pretty low reproductive barriers between species. It's important to kind of also understand that your unimodal hybrid zone can look something like this, where you have more hybrids in your zone than either of your parental species or parental types. All right, so we will talk about the study system. I'm working within the americanus subgroup, and that's composed of these 4 species: americanus, Sansinae, cubae, and bulbopes. And then there's another one, Habronattus Y, but that's not included here, or really any of my other studies in this subgroup. But what makes this particular group interesting is the number of different forms that each of these described species has. So americanus here has these 4 different forms, Sansinae has 2 here, cubae has 2, and bulbopes. And there are probably certainly more than just these forms listed here. So there's a lot of morphological diversity. And then, of course, there are additional more isolated forms that don't seem to fit in with any of the described species. So pavant, Sansinae, cedar city, and gunnison, these don't really seem to fit in with any described species. And there are definitely more than just these 3 sort of unique morphs. Right. So in a previous study, we looked at how hybridization and introgression combined with the divergence history has affected the evolution of the group as a whole. So basically, we found that the genomes seem to be extremely homogenized. So just as an example, in this UCE phylogeny, the different colors are representing the different morphs or the different species. And you can see that the different colors are basically spread out through the entire phylogeny. So rather than our evolutionary lineages reflecting species identity, they're more corresponding to this geographic location. And that's another signal to reiterate the degree to which the genomes are homogenized. We also found considerable morphological diversity. So these are all of the different morph types that I could sort of accurately define. And the genomic data only supported 3 sort of geographically clustered or centered clusters. So there are a lot of different morphs and species within each one of these genetic clusters. Again, just kind of showing how homogenized the genomes are despite what the males look like. Okay, so a couple big sort of main takeaways from that whole subgroup study were the high morphological diversity despite this evidence for widespread gene flow across the group, and our highly homogenized genomes across all of the different species. And then all of this together kind of suggests that there's a small number of loci that are responsible for the courtship traits and species identity within the group. Okay, so when we think about hybrid zones within the group, the most common hybrid zones are between americanus and cubae, and sometimes there's another species. So up in Ashland, bulbopes is also involved. But these blue markers here are just examples of hybrid zones that I've visited and collected. For this specific talk, we're focusing on a hybrid zone that happened at Mount Shasta. And this is a hybrid zone between americanus P morph and the cubae north morph. So using data from that specific hybrid zone, we wanted to characterize the genomic architecture of the hybrid zone, describe the morphological diversity within the hybrid zone as well, and then see how these things in common might act as a microcosm of the broader evolutionary dynamics of the whole group. Okay, so this is Mount Shasta from the south side, and these are just some of the common landmarks that you'll see if you're looking at it from the south side. And here is Panther Meadows at an elevation of about 2,286 meters. And this is where the hybrid zone site or collection site is. If we think about the last glacial maximum during the Pleistocene, a glacial advance covered this south side in ice down to about 1,920 meters. So that's obviously lower than the hybrid zone location. So this was covered in ice sometime between 17 to 11,000 years ago. So the oldest that this area or this hybrid zone could be is between that 17 to 11,000 years ago timeframe. Okay, so we went out to collect these spiders in this hybrid zone, and these black dots are just where we collected all of the individuals from, kind of ranging up and down the zone. We ended up with 99 total specimens collected, with 78 males and 16 females. And naturally, for the americanus subgroup, the diversity of spiders that we found was a little bit crazy. So there were a lot of different hybrid types and diversity within male morphologies. So the types of traits that they shared among both parental types was remarkable. So we brought them back to the lab and we scored 14 morphological characters for all of the males, right? The females don't have these colorful ornamentation or anything like that. So this is just an example of some of the traits that we scored. So things like color, striation patterns, what the pattern of the iridescent clypeus looked like, and so forth. So we have both of our standard or parental species types, americanus here, and then cubae here. And then we had 19 distinguishable hybrid forms. So there's a lot of diversity within the hybrid types and the traits that they're combining between both parental types. And I think it's worth noting, or a little bit interesting, that the white palps here, the hybrids that have this white palp trait, that's a novel trait. Neither parent species has this pure white palp form. That's kind of neat. There's something weird going on there. And since there are so many hybrid types, 19, to make visualization a little bit easier, I broke them down into types of palp color. So like red hybrids are in green here, yellow hybrid palps are in light blue, and then white palp hybrids are in dark blue, and then our parents, so black for americanus and red for cubae. So we wanted to see what the morphological diversity looks like. Just to visualize that, we ran an NMDS plot. And if we look at this, it kind of makes sense. So we have a separation with our parental forms, are a standard type, the americanus down here in black and cubae over here in red. And then in between are all of the different hybrid types. And that makes intuitive sense since our hybrids are sort of a combination of both of these parental types. So they're sharing traits from both parent. Okay. And the spatial distribution of the hybrids and the parental type across the zone is pretty even. So there isn't really a pattern that you can identify. It doesn't seem to be organized as far as, morph type or anything like that. There's not cubae down at the bottom and americanus up at the top and so forth. And so because of this, we kind of categorized this as a unimodal hybrid zone. But probably it seems like there might be more hybrids than actual parents in the population.
Yeah,
all right.
That's 10 minutes, Tierney.
Okay, thank you. So we did some RAD-seq data too, and we collected about almost 1,600 loci with this min-SAMP value of 90% and ran things through using stacks. A structure analysis came through at a best K value equal to 2. So the sections here are divided by those same hybrid morphology types and then the parent types. So you can see that all types share ancestry from both the blue and the orange clusters at pretty much equal frequencies. And just a note, the pure populations here, these are populations that are near the hybrid zone, but they're not actually within the hybrid zone. So these individuals should theoretically have pure genomes. But the main takeaway is that it's pretty difficult to find any structure here that's correlated to hybrid type. So maybe just to see if there was any geographical structure across the zone, we divided this longitudinally. So the west side over here and the east side is over here. Again, there's not really any pattern that you can pick up on. The types seem to be spread out throughout the both sides, and they're sharing the same amounts of blue and orange genetic ancestry. So maybe longitudinal or latitudinal geographic structure exists, but that also does not seem to be the case. So it's still kind of just nothing that we can see. All of these types, no matter how we divide it up, have both types of genetic ancestry. Okay, so to reiterate the lack of genetic structure, we ran this PCA, and it's just visually looking at this, you can see that there really aren't any distinguishable groups. So it's just kind of a blob of phenotypes, including our pure populations. So really a lot, like very genetically identical. Okay, so in summary of what we found here, we have extreme morphological diversity in hybrids, 19 distinguishable forms plus 2 parent forms. And then our hybrid and standard forms are present at equal distributions across the zone. So individuals are nearly genetically identical, at least with the RAD data that we collected, and there's no structure according to hybrid type. These findings fall in line with this bigger study of the subgroup as a whole. So strong selection maintaining male morphologies despite genome homogenization, and small loci responsible for morphological differences between species. So a running hypothesis that we have is the americanus subgroup individuals speciated along color lines followed by introgression within geographic regions. And this kind of makes sense and perhaps is promoted by americanus into others when these come in symmetry. So maybe for some reason this red morph is favored over the yellow or other palped colors, since we have more red phenotypes in the population. And it's really the kind of cool thing about this is we have a great group to understand, and it's really encouraging to identify the genetic basis of courtship traits, like what they do with a lot of bird studies. We obviously need to figure that out a little bit more and get genomes, but it would be a really cool thing to be able to make a connection to the genetic loci that are corresponding to these male color traits. Okay, so I'd like to extend a thank you to all these people and permitting and of course, everything like that. And I can take questions if I have time.
Thank you so much, Tierney. I'm sure everyone is clapping, although we cannot hear them. And we have time for a couple of quick questions. So Brian asked for the NMDS graph, what percentage of variation is explained by each axis?
Like an exact answer for that. Mostly just visually looking at this is kind of all that I have for that answer, but thanks. I can look into that more.
Okay. Then Eileen Havets asked, why do you think you found so few females compared to males?
Yeah, that might just be the timing of collection and the timing that we're out. So it's also easier to see males because they're really colorful. Females are hiding a little bit more. The males are running around trying to find, a mate. But females are a little bit more cryptic to find, and that's probably what that is from.